Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 4 for June 2020. This is the part 1. I will do this paper in two parts, part 1 as well as part 2, and I'll put the link to part 2 in the description box below this video. Let's begin. Question 1 says, chloroplasts are involved in both the light-dependent reactions and light-independent reactions of photosynthesis. They ask which row of the table shows where the light-dependent reactions and the light-independent reactions take place. So we need to know, since these are light-dependent reactions, they need to occur in a place that has the chlorophyll, which is the pigment that traps light. And since chlorophyll is located within the thylakoids, we can say this is good. The light-dependent reactions will occur in the thylakoids, and the light-independent reactions are going to occur in the stroma because they do not need the raw light. So basically, the diagram shows the outline of a chloroplast. They say draw 3 labeled features on this diagram that are found in a chloroplast other than the stroma and the thylakoid membrane. So the structure was drawn and the membrane was labeled, but there was no inner membrane, so I drew the inner membrane and labeled it. And then I drew the thylakoids as well as the connecting membrane, which is the lamella. I put in the DNA as well as the ribosomes. I labeled the granum, and then the stroma here and the starch grain. So the things we were looking for was maybe starch grain, the ribosome, the DNA, and the granum, because the rest were already, they told you not to put them, they were not necessary basically. So any of these, you'd get the 3 marks you're looking for if you had put 3 of those. I marked with a specific marks, you can see that. So moving on, an absorption spectrum shows how much light is absorbed by chloroplasts at different wavelengths of light. The table shows the color of light at 4 wavelengths. So here we have the wavelengths, 460, 520, 600, and 680, and their corresponding color of light. They ask which wavelength of light is absorbed the least by chloroplasts. Now, chloroplasts appear green. That is because the green color is not going to be absorbed. So it's going to be that wavelength because of the unabsorbed green light color. So it's going to be 520 nanometers. The next part says, state what is meant by the term action spectrum. An action spectrum is a spectrum. It shows the rate of photosynthesis at different wavelengths of light. So as I can show you the example I put here, this is an example of an action spectrum. On the vertical axis, you can see rate of photosynthesis, and the horizontal axis, we have wavelength. So it shows you the rate of photosynthesis at specific or at different wavelengths. Chloroplast pigments can be separated and then identified by their RF values, which process can be used to separate chloroplast pigments. Because they've talked about the RF value, you automatically know this should be chromatography. So the answer is A. However, you need to know the other definitions. As you can see, dendrochronology studies tree rings or growth of tree rings. Osmosis is the movement of free water molecules from a region of high water potential to a low water potential region. And PCR amplifies the number of DNA molecules. So the only correct answer here was chromatography. Let's move on. The diagram shows separate chloroplast pigment J and K. So we can see there is the solvent front here. There is the baseline of what you call the origin. And then we can see there is a direction of movement. So we have K and J. They ask what is the RF value of chloroplast pigment J. To find the RF value, you need to divide the distance moved by the pigment from the origin or from the baseline by the distance moved by the solvent from the baseline to the solvent front. So we have to measure from the baseline to the point where the pigment is, and from the baseline to the solvent front. Divide that distance by that, and you will get the RF value. In this case, I have a 6 divided by 7.5, and I get 0.80, which is going to be answer B for my RF value. This brings us to the end of question 1. Let us go to question 2. Body temperature and the degree of muscle contraction can be used to determine the time since death of a person. The table shows how body temperature and body stiffness due to muscle contraction change with time since death. So we can see body temperature and body stiffness. Of course, we expect the body not to be stiff, then stiffen, and later loosen as decomposition begins. So at this point, we see decomposition has begun. That is why the muscles are not going to be stiff. Here, it's because they are in rigor mortis due to loss of ATP. So the body temperature is also going to be decreasing. So let us see. They say, state how the temperature of a dead body should be measured. You can measure using, maybe you use the lever, or you can use a rectum. Use a rectal thermometer to measure the rectum, or it should be in the liver or deep within the body. Any organ deep within the body, that can be used. So you will insert the thermometer and take the temperature, which is the core body temperature, could be in the rectum or in the liver. So here the next part says the body temperature can be used to estimate the time since death using the following information. Loss of 0.78 degrees per hour for the first 12 hours after death, and 12 hours loss of 0.4 degrees Celsius per hour. That is after 12 hours. Estimate the time since death of a person whose body temperature had fallen 11.5 degrees. So remember, for the first 12 hours we have this, and for after 12 hours that it's going to be dropping by that per hour. So let's see. Remember, they want us to show our answer to the closest hour. So the loss in the first 12 hours is going to be, since this is per hour, that times 12, which gives us 9.36. And you can see 9.36 and 11.5, they're another same, so there is a balance. To get that balance, we say 11.5 minus 9.36, which gives us that. Now we know this is going to be in the next 12 hours. So how do we find how long it took? We need to divide this by 0.4, because 0.4 is a drop per hour. If it dropped by 2.14, divide by that to find it's 5.35 hours. And when we add that to the 12, we get 17.35 hours. So this is approximately 17 hours. So that is the answer, should be 17 hours. Remember, they said nearest hour, so we round off to that. Next part. Explain why this estimate would be different if the body had been left in a colder place. This is self-explanatory, because if the body had been left in a colder place, the heat loss would have been faster, and the cooling time would be shorter. That would be because the person or the body is losing heat to the environment due to a lower ambient temperature within the environment. So this will make the estimate to be shorter than the calculated value. The next part says, explain why using body stiffness only as shown in the table is insufficient to estimate the time since death accurately. You guys saw there is stiffness initially. Actually, it's not going to be stiff, then it stiffens, then it's not stiff. So if you find the body not stiff or stiffened, you could misread the results, because body stiffness, which is rigor mortis, can be affected by temperature. And sometimes deciding whether the body is stiff or not stiff could be subjective, because, one person could see it as being stiff and another person could see it as maybe not stiff enough or not stiff. So that could be subjective depending on the person or the viewer or the person who is examining. So based on the table, we can see that using body stiffness can only be used between 3 to 36 hours. And again, to take you back, so we can see before 3 hours, it's going to be not stiff, then that, until here. If you find the body not stiff again, it could be because it's decomposing, and that will not be sufficient information to let you predict the time of death properly. So here I say 3 to 36 hours also. So if the body is found before 3 hours or after 36 hours, we cannot only use body stiffness to predict the time of death. And again, to take you back, remember, less than 3 hours is not going to be stiff, so we can only use within this time period from 3 to 36 hours, because it's going to be stiff, it's going to be stiff. It means if we ever find that body to be stiff, it is going to be within this range. When we find the body not stiff, it could be in this range or it could be in this range, which could be misleading, so we can only use within that time frame. So this brings us to the end of question 2. Let us go to question 3. Question 3, the following equation summarizes photosynthesis. So there is 6 carbon dioxide molecules, 6 water molecules, giving us glucose, and 6 oxygen molecules. They say the diagram shows the bonds in carbon dioxide, water, and oxygen. So we can see them here. And then they ask, energy is needed to break the chemical bonds and to form new chemical bonds. This is called the bond energy. The table shows some bond energies for the bonds in carbon dioxide, water, and glucose as well as oxygen. So they say the type of bond. This is information in the table, the types of bonds and their bond energies. So they ask, in photolysis, one of the bonds in each water molecule is broken. Remember, they've said one of the bonds. Using the equation for photosynthesis, calculate how much energy is released by photosynthesis in order for one molecule of glucose to be made. Now, what we need to know is, since one bond is broken, only one bond, and you remember we have many of those. Let's take you back. For water molecules, we're going to have 6 water molecules. So if you're breaking just one, and the energy required to break the oxygen-hydrogen bond is 462, it means we're going to have 6 times 462 to find the energy that is going to be required to break those bonds. And again, remember, the energy to break part this to that is 462. So 6 times the 6 water molecules will be that times 6, giving us that, which is going to be the answer here. So let's continue. Explain how light energy is converted into chemical energy in the form of ATP. Now, in this case, we're going to talk about how light is going to be used to, is going to be absorbed by the chlorophyll molecule, and then the electrons excited, they move through the photosystems, and then ATP is generated. So here I say light is absorbed by chlorophyll in the photosystems, and the electrons are excited from the chlorophyll molecules. So these are released to higher energy levels, and as the electrons are passed through the series of electron carriers, this is within the photosystems, or as they move within the cycle, the cyclic and non-cyclic photophosphorylation. So in this case, as they move through, as they are passed along the electron carriers, so carriers are arranged at different decreasing energy levels. As you can see, one is going to be at a high energy level, the next one is going to be at a slightly lower, and as the electron moves through those decreasing energy levels, or the carriers positioned at the different energy levels, we're going to see energy being lost, and that energy is going to be used to create ATP. So I said the movement of electrons through the carrier molecules releases energy, and this energy is used in the phosphorylation of ADP to form ATP. So phosphorylation is going to occur between ADP and Pi, leading to the formation of ATP. The key thing here is knowing that light splits, or light causes the electrons to be excited from the chlorophyll molecule, and then they move to higher energy levels, and then they move back through the electron carriers that are positioned. So they move back through the electron carriers that are positioned at different energy levels, and as they do, energy is going to be lost by those electrons, and that energy is going to be used to make ATP. Let's move on. So here they say the diagram shows the structure of glucose. So we see the structure of glucose, and then they ask, the energy needed to form the bonds in one molecule of glucose is 9164 kilojoules. State what other information is needed in the table of bond energies for this value to be calculated. If we want to calculate this value, remember these are all bonds formed. So if they are bonds formed, we have formed the oxygen-hydrogen bond, but we need to form the carbon-hydrogen, carbon-oxygen, carbon-carbon, all these bonds. Some are going to be broken in the process and others are going to be reformed. So we need to be able to know that. Remember, the carbon-oxygen bond is broken in carbon dioxide, and the carbon-hydrogen bond, carbon-oxygen, carbon-carbon bonds are going to be formed, as well as oxygen-hydrogen. So that will require some energy, or you can be able to use that to calculate the energy that is required. So here they ask, where in the chloroplast are these bonds formed? The bonds are going to be formed in the stroma. In the stroma, because that's where the dark, or I would say the light independent reactions occur. Please do not say dark reaction, just say light independent reactions are going to occur in the stroma. So that is where glucose is going to be made. Glucose is used by plants in the production of amino acids. Which row of the table describes how amino acids join together? So I'm going to use an example here. Let me use a different color so that you can see properly. So I'm going to use a typical amino acid, which could be that C. There is an R group here, and then a hydrogen, and a C oxygen, or H. And then on the other side, let me use the same thing, H, N, that is H, C. So we have the R and H here, and the carboxylic group the other side. So if this is to combine, this hydrogen here and this OH are going to be converted into water, and then we'll have H and H, and then Cr hydrogen here, and the C double bond O on the other side, linking to the nitrogen with a hydrogen, and the Cr, there is an H here, and the COOH. So this is the bond linking the two specific amino acids. So the bond should be between a carbon and nitrogen. And the type of reaction is condensation, because water molecule is lost. So the answer here should be an A, carbon and nitrogen, and condensation reaction occurs. The next part says, explain why amino acids cannot be produced from glucose alone. So the thing is, glucose will only contain carbon, hydrogen, and oxygen, yet in the production of amino acids, we need a nitrogen atom, and sometimes we need a sulfur atom. So glucose only contains carbon, hydrogen, and oxygen. However, amino acids contain carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. So the nitrates will provide the nitrogen, and sulfates could provide sulfur. So we can. That's basically how you can reason this. This brings us to the end of question 3. Let us move on to question 4. Rheumatoid arthritis is an inflammatory condition caused by the overactivity of the immune system. The photographs show the hand of a person with rheumatoid arthritis and the hand of a person without rheumatoid arthritis. So you can see this person here has and that one doesn't have. So they say, give one piece of evidence shown in the photograph that rheumatoid arthritis is an inflammatory condition. We can see there is swelling. Inflammation can lead to swelling, so we can see there is swelling. So the swollen hands show that there is inflammation. And then they say, give 2 other signs of inflammation that are experienced in the hand of a person with rheumatoid arthritis. There should be pain. You remember when you talk about inflammation, there is swelling, there is pain, there is redness, there is increase in temperature or increase in heat. So all these can be symptoms of any inflammatory condition occurring. So these are the possible answers. Let's continue to the next part. It says, tumor necrosis factor, or you could say TNF, is a protein that can be released by several types of cells. It plays an important part in the immune response. The protein binds to specific receptors on the surface of cell membranes, stimulating chemical reactions inside the cell. As a result, a number of responses may occur that include inflammation and the stimulation of phagocytosis by macrophages. Antibodies of TNF are used to treat rheumatoid arthritis. Scientists have used a virus that infects bacteria to produce antibodies to human TNF. The diagram summarizes this process. So here we see the gene of human TNF is attached to the viral gene that codes for the protein in the capsid. And then the virus replicates and TNF is displayed on the outside of the capsid. And these viruses are used to make antibodies to the TNF. So they explain how attaching the gene for TNF to the viral gene results in TNF being displayed on the outside of the capsid. So here I said when the virus replicates, remember there is going to be replication, the viral genes containing the human TNF gene will be transcribed as well because the DNA is there. So they'll be transcribed and translated into the protein. Now the protein is going to be TNF, which is generated. And then during the viral reassembly, as the virus is being reassembled, TNF will be incorporated into the capsid of the virus. Remember, after a virus infects a specific cell, it's going to use the cell to replicate itself, creating more genetic material of itself, and then creating proteins that are structural proteins for surrounding the genetic material. And in the process, there is going to be the transcription and translation of the TNF protein, or you could say TNF synthesis, and that could lead to the incorporation of this specific protein in the capsid of the virus. So let's continue. They say, explain why antibodies to TNF are used in the treatment of rheumatoid arthritis. This is asking you about how antibodies work. So the antibodies will bind to the TNF, causing agglutination. Remember, antibodies cause agglutination. They can cause, they cause in agglutination, they can cause them to stick together, and then they could cause so many other the processes that can lead to the destruction of the specific pathogen. So when they cause agglutination, they prevent the TNF protein from binding onto cells, because that means the substances containing this protein are going to stick together, and therefore they cannot bind to the new cells, or that the rate at which they bind to the new cells is going to be reduced. So therefore inflammatory responses will not be triggered. So if they are not attacking the new cells or binding onto the proteins, it means binding onto the cell membranes, it means then inflammation will not be triggered, and that will relieve the symptoms of that. They're going to say patients being treated with antibodies to TNF are more susceptible to tuberculosis, which can be fatal. Explain why patients being treated with antibodies to TNF can die from TB. Now let's see here, because you are using antibodies to TNF and this bind to the TNF, so phagocytosis and macrophages will be reduced. You can understand that, and as a result, there will be fewer fewer bacteria will be destroyed, because remember, tuberculosis is a bacterial disease, or it's caused by bacteria. So if the macrophages and the phagocytes are reduced, or I would say phagocytosis by macrophages is reduced, it means fewer bacteria are going to be destroyed by these macrophages or phagocytes. And since the antigen presentation by the T cell will be hindered. So remember, the T cell gets its information or is triggered by an antigen presenting macrophage. If the macrophages are not presenting their antigens, it means the T helper cells will not be triggered. And if they are not triggered, it means they will not cause the B cells to rapidly divide, causing the production of antibodies. So here, consequently, we can see the tubercles will form, destroying the lung tissues, leading to organ failure, and finally to death. And the key thing you need to know here is, if we have fewer phagocytes or fewer macrophages presenting antigens to the T cell or causing the T helper cells to be formed, it means we will have fewer T helper cells, will have few activated B cells or B plasma cells, and therefore we will have fewer antibodies being produced, and that will lead to organ failure, because the tubercles will become more and more, destroying the lung tissues. So this brings us to the end of question 4. Please do not forget to subscribe to our channel. I will see you in the next video when we talk about part from question 5 to question 8. That will be the part 2 of this series. So bye-bye.